Hi, and welcome to our webinar today on extrapolation of foreign data for U.S. marketing authorizations. Our goals today are to share with you some of the key factors involved in successfully using clinical trial data generated outside of the U.S. to support NDA and BLA applications. Just a reminder as we go through today's presentation, if there are any questions, please put them in the chat. We'll do our best to address them at the end of the presentation, and for anything we don't get to, we will follow up afterwards. For our conversation today, I'm pleased to be joined by one of my talented regulatory colleagues from Quantum Regulatory Solutions. I will start by introducing myself. I'm trained as a pharmaceutical science and have been working in the industry for more than 28 years. Along with Mark Amon and Mara Hollinger, I co-founded Quantum Regulatory Solutions in May of 2023. I've worked on both the sponsor and consulting sides of the business with several years in international senior leadership, including managing regulatory affairs, project management, drug development, and business development teams. I have extensive experience with regulatory due diligence, assessments supporting both sellers and investors, and excel at building client relationships and solving complex problems by deploying proven strategies to overcome regulatory and business hurdles. Kevin?
Thank you, Dawn, and hello, everyone. My name is Kevin Hennegan. I'm head of advanced therapies for Quantum. I have over 20 years of regulatory affairs experience in the pharmaceutical, biotech, and medical device industries. And although I have supported clients in a variety of areas, I have particular expertise in vaccines, cell therapy, and gene therapy. I also have experience in all stages of drug development and in multiple clinical indications. I have my master's degree in molecular, cellular, and developmental biology from the University of Colorado at Boulder.
So our topic today is centered on the use of data from studies conducted outside the U.S. to support U.S. marketing applications. Over the past few years, there have been several examples of NDAs or BLAs that heavily, if not exclusively, relied on foreign data. And this has provided the backdrop for FDA to put forth their current thoughts and opinions on these situations. In anticipation of its advisory committee meeting for sintilimab in 2022, the FDA released a briefing document focused on standards for single country foreign data submitted to FDA for authorization in the US. The document outlined and established several new requirements for use of foreign data as the sole basis for marketing approval. Those requirements were subsequently codified into regulations. Specifically, the data must be applicable to the US. The studies must have been conducted by qualified investigators, and FDA must be able to inspect and validate the study data. These requirements expand on the previously established principles for FDA accepting data from foreign studies as part of a development program and are further elaborated in guidance documents to include additional provisions, such as requiring GCP compliance and allowing sponsors to request waivers from standard IND requirements. A couple of the most common waivers requested include specific IRB composition requirements and collection of signed 1572 forms. Now, Kevin will provide a little more detail on ICH E5.
Thank you, Don. So the ICH E5 guideline provides the global framework for relying solely on data from other countries, or a combination of data from other countries and bridging studies within the new second region. ICH released E5 in early 1998, and FDA released an associated Q&A document in 2006. The premise of the ICH E5 guidance document is that it is desirable to reduce duplication of clinical studies. Trials are expensive and, in some situations, ethically inappropriate to repeat when the safety and efficacy of a drug have already been demonstrated. But that goal must be balanced against the risks of ethnic or regional differences impacting the safety, efficacy, or dosing of a drug. So we'll talk a little bit more about that today and what's necessary in order to pursue an ICH E5 approach to support approval of a drug in the US on the basis from trials conducted in a separate region.
Kevin, sometimes when we talk to clients about this extrapolation approach is seen as maybe a shortcut or a way to apply foreign standards to US filings. Is that really the case?
No, not really. It's a shortcut only in the sense that it may reduce the amount of data that you need to collect within the US. All of the other requirements of your data package, including GCP compliance, trial design, statistical rigor, all of those remain in place. So ICH E5 recommends criteria for assessing when those requirements might be satisfied with your foreign data. In cases where those criteria are not met, the guidance goes on to make recommendations for clinical bridging studies or additional full clinical trials that could support extrapolation from the foreign region to the US.
Great. Thank you.
Absolutely. So let's go over a few definitions that are specific to ICHE5. The first is ethnic factors. Ethnic here is broader than just race and ancestral ethnicity, the way that some of us might typically think of it, in that it includes any common traits and customs shared across a large population group. Ethnic factors are subdivided into intrinsic and extrinsic factors. Intrinsic factors includes those racial and ancestral ethnicity elements, but also include things like genetic disorders, age, body weight, and organ function. Extrinsic factors, on the other hand, include environmental and cultural elements from the individual's location. This includes things like local medical practice, available therapies, diet, climate, socioeconomics, disease definitions, and GCP practice. So finally, let's define a bridging study, which is a supplemental study usually conducted in the new region that will collect PK, PD, safety, or efficacy data to support extrapolation of data from the foreign region to the new region.
Kevin, how's the best way to get feedback on whether a bridging study might be needed? And if it is, what data needs to be collected in that study?
Yeah, it's a good question. It's my opinion, it's always a good idea to engage FDA in a meeting when you think there's an appropriate amount of information known about the drug to have an informed conversation about it. So this could be as early as a pre-IND meeting if the sponsor has some information and knows that they are planning to conduct a significant portion of their clinical development program in a foreign region. The sponsor should also be doing their own evaluations of the program based on some of the concepts we'll go over in the next slides. So now that we've defined a few of the terms, let's talk a bit about the criteria that ICHE5 recommends for extrapolating clinical data from one region to another. And the first element is that you need to start with a complete clinical data package in the foreign region where the drug has been tested. This includes a comprehensive assessment of the drug's pharmacokinetics, pharmacodynamics, and dose response, as well as the drug's safety and efficacy. You should also usually characterize PK, PD, and dose response in a population that is representative of the new region. Now, that doesn't necessarily have to be done within the United States, for example, but it should be in a population that is representative of that region. If a drug is ethnically insensitive, and we'll talk a bit about what that means in a minute, that may be all that you need to support extrapolation. However, before agreeing to an extrapolation approach, FDA will assess the pivotal clinical studies proposed to support the marketing application to ensure that it meets their regulatory standards for approval. And some of the basic issues to be considered in a study design that could affect FDA's willingness to rely on those data include the definition and diagnosis of the disease condition and of the patient, ensuring that what you are targeting to treat in the US is consistent with how that was defined in the clinical studies you conducted elsewhere. Your choice of control group, your regional target or objective of treatment as that relates to your efficacy variables for the study, your methods for assessment of safety, medical practice, the duration of the trial, any regional concomitant medications to ensure that those are consistent and don't change the context of your clinical data, severity distribution of eligible subjects, and similarity of the dose and dose regimens for what you will be proposing to use in the US.
Kevin, many of these factors will be context and essentially program specific, but are there any clear lines between when FDA would insist on having bridging data versus when FDA would be willing to agree to approve an application based solely on the findings from the ex-US data?
Yeah, there aren't a lot of clear lines, as you say. These are largely context dependent, but I think there are a few, and I think one example of that type of line is when the regional prevalence of a disease is so low in the U.S. that it makes clinical studies impractical here. So tropical infectious diseases like malaria or dengue fever are good examples of this. We'll discuss another example later in the presentation when we review other examples of applications that have utilized the ICHE5 approach.
Great.
All right, so FDA's decision tree for assessing the acceptability of a foreign clinical data package is illustrated in this diagram, which is adapted directly from the ICHE5 guideline. And the first question that FDA will assess is whether the clinical data package fulfills all US regulatory requirements. If the answer to that question is no, then additional clinical studies that go beyond the definition even of a bridging study will be needed in order to fulfill those requirements. That portion of the decision tree is actually not presented here as it's beyond the scope of our webinar today. But if the answer is yes, then FDA will go on to assess whether extrapolation of the data to the US population is appropriate. If the answer to that question is also yes, then no further clinical study is needed, and you should have a clear path to a marketing application, at least with respect to clinical data. If the answer is no, then a bridging study of some type will likely be needed. So let's talk about how to go about assessing the data needed to answer that second question. And that assessment begins with an assessment of the drug's sensitivity to ethnic factors with respect to its PK, safety, and efficacy. A drug that has linear PK, a wide therapeutic dose range, and little potential for drug-drug interactions will usually be less likely to be sensitive to ethnic factors. Examples of this include things like monoclonal antibodies and corticosteroids. On the other hand, drugs that have a steep pharmacodynamic curve, that are metabolized by a single polymorphic pathway, or that may be used in a setting with multiple co-medications, are more likely to be ethnically sensitive. And some examples of drugs with known sensitivities in those domains include things like statins, ACE inhibitors, and antidepressants. characterization of a medicine according to the potential impact of ethnic factors on its PK, PD, and therapeutic effects can be useful in determining what sort of bridging study is needed in the new region. The impact of ethnic factors on a medicine's effects will vary depending on the drug's pharmacologic class, the clinical indication, as well as even the age and gender of the patient.
Yeah, Kevin, are there any particular factors that a sponsor should give more or less weight to in this evaluation?
Was no one property of the medicine that is fully predictive of the compound's sensitivity to ethnic factors. You should really check all the boxes for the factors on the less sensitive side. If you have any of the items on the more sensitive side, then you may well need a bridging study. The type of bridging study is ultimately a matter of judgment, but the overall assessment of sensitivity to ethnic factors may help in that assessment. So let's go on and talk a little bit about the differences between the intrinsic and extrinsic factors. So if you are pursuing an ICH E5 strategy, you should conduct an assessment of your drug's ethnic sensitivity that considers both intrinsic and extrinsic factors. Things like in vitro drug-drug interaction studies are part of that intrinsic factor assessment. Drugs metabolized by enzymes with known variability across ethnic subgroups are more likely to be sensitive to those intrinsic ethnic factors. Uh, Evaluation of intrinsic and extrinsic factors should focus on how the factors may affect the performance of the investigational product. However, factors that influence the disease in domains like severity, prognosis, or susceptibility should also be considered, as if there are significant differences between regions that could influence the confidence FDA has in those foreign clinical trial results. Some of the intrinsic factors that will need to be reviewed are comorbidities. Is there a coincident disorder that increases your risk or severity of drug side effects, for example? Age and sex should also be evaluated for any impact on safety or efficacy. And FDA expects sponsors to assess their drugs for impacts of renal and hepatic insufficiency. Some of these assessments you may be able to complete through subgroup analysis of your foreign clinical trial data. Age and sex are pretty obvious candidates for that, while others may be assessed through in vitro or in vivo toxicology studies. For extrinsic factors, some key elements to consider are the disease definition. As I said earlier, are you treating the same condition, or are there local differences in diagnosis or nomenclature that result in significant variability? We've had some recent experience with this, and we'll discuss that further later in the presentation. Therapeutic approach has been a major, well-publicized issue as it pertains to assessment of extrinsic factors. Differences in the availability and use of other therapeutics for a clinical indication alter the overall context of your clinical data and have resulted in FDA refusing to approve drugs like sintilimab, for example, solely on the basis of ex-US data. The local standard of medical practice can have a significant influence on treatment outcomes, particularly for indications that have a long treatment or recovery period. So ensuring a consistent standard of supportive care is important to support generalizing data from one region to another. And finally, local quality of regulatory practice or GCP compliance can have a significant influence on FDA's willingness to rely solely on foreign data. The availability of data for inspections is an important part of that, but it's not the only factor. So depending on the ethnic sensitivity of your drug and the overall context of the program, different types of bridging studies may be needed. As we have mentioned previously, it's good to have a plan for what you think is needed from these bridging studies and then to run that plan by FDA to get their buy-in. I should note that bridging studies can be done at any stage of development and can be performed to collect different types of data. Most commonly, PK/PD studies may be done to determine if the exposure and kinetics of your drug are consistent from one population to another. If there are differences, dose response studies may be needed to better define the dose adjustments necessary for the new region for the US. Bridging for safety may be necessary when there's concern about the generalizability of the reported adverse events to the US population. There is evidence in the clinical literature that patients outside the US report AEs less frequently than American patients do, though the extent of that difference can vary by clinical indication. Based on my experience, this type of bridging is unlikely to be needed for orphan drugs and rare diseases, but may be necessary for conditions with a larger patient population. Efficacy bridging may be necessary even in rare disease in situations where there's a higher risk of sensitivity or where there are other concerns about the quality of data from the foreign region. When bridging for efficacy, randomized controlled clinical trials are preferred, as they are throughout clinical development, but these do not necessarily need to duplicate the foreign pivotal trial. A shorter study using an alternate or a surrogate endpoint may be sufficient to provide justification for extrapolating results.
Do we have an example of FDA acceptance of a surrogate endpoint for bridging of efficacy?
Yeah, it's a good question. That factor is specifically mentioned in the ICH-5 guidance, but I'm not aware of a product yet that's been approved based on a surrogate endpoint for bridging of foreign clinical trial data to the US. Um, Approval of Sarepta's gene therapy, Elevidys, for the treatment of Duchenne's muscular dystrophy was based in part on a bridging study, but in that case, they were bridging manufacturing processes from from one process to another, and they used a surrogate endpoint of expression of microdystrophin to demonstrate that bridging element. Uh, Also worth noting that that particular approval was only granted after an override of the review team's recommendation by the former CBER director, Dr. Peter Marks.
Kevin, one factor you mentioned that does need highlighting is the agency's familiarity and experience with the countries or regions used to generate the ex-US data. And most of the time, this is their familiarity with the country or region's compliance history. If you look at FDA's historical inspection records, there are some data that show a higher level of non-compliance in certain countries. I think India and China have fallen into those categories as well as other countries, but both India and China have made a lot of progress over the past couple decades, also in trying to implement new quality systems for their clinical studies. But it's good to be aware of this so that the sponsor can work to mitigate concerns prospectively when the majority of your data may be coming from those countries or regions. There's equally the possibility of the unknown too. If it's a country where FDA has not seen a lot of clinical data generated from that specific region, that in itself may also cause some hesitation where the agency is unfamiliar with local practice and or local compliance in a given country.
Yeah, I would say that that's true based on my own experience, though I think it is important to note that it's not just a socioeconomic factor. Clinical trials have been conducted to a very high standard in a number of low-income countries in Africa, Southeast Asia, and South America, and have supported U.S. approvals. Approval of the Ebola vaccine, for example, was supported by trials conducted exclusively in Africa. Um, I will say that I have some concern that some of the changes in U.S. policy positions may have impact in this area, which, whether due to the potential for restricted access to site inspections, or within FDA, an increased preference for U.S. data. Although I think it's probably too early to tell yet if those risks will become realities, but I think it's something for all of us to keep an eye on. All right. So as we consider the use of foreign clinical trial data, we thought that it would be important to also briefly discuss how this ties into ethnic coverage of your clinical program and FDA's guidance on diversity action plans. In raising this issue, we want to acknowledge the elephant in the room. And in fact, FDA's guidance on diversity action plans was briefly taken down after the change in administration in January. However, as of the date of this webinar, the guidance is back up, albeit with some interesting caveats on the FDA website. Um, and we believe that a diversity action plan should still be a part of most development programs. There are classes of drugs that are known to perform differently in patients of different races, and gender differences are also known for drugs for like opioids and antidepressants. So the ICH E5 guidance recommends characterizing PK and PD in at least the three major racial groups relevant to the ICH regions, so Caucasians, Blacks, and Asians. And expanded evaluation in a particular race may be needed based on your own risk assessment. Multi-regional clinical trials can improve the diversity of enrollment in your study, but you will still want to make an effort to ensure enrollment of adequate subgroups representative of US epidemiology for your clinical indication. There we go. So let's go through a few examples where FDA has granted approval based solely on ex-US data. Artesunate is an anti-malarial drug that received approval in 2020. And the rationale for use of foreign data for approval in that case, as we discussed earlier, is that malaria is not endemic in the US. So that approval was based on data collected from nearly 7,000 patients who participated in controlled clinical trials conducted in Africa and Southeast Asia. Radicava is a drug approved for the treatment of amyotrophic lateral sclerosis. It was approved in 2017. That approval was based on data from 368 participants in controlled clinical trials that were conducted in Japan. The product had actually been approved in Japan before the NDA was submitted to FDA, so FDA actually had no input into the development program. Nevertheless, the drug was approved based in part on an ICHE5 analysis that found low risk of impact from intrinsic and extrinsic factors. Population PK studies, which did include data in Caucasian patients, included no differences in PK due to race, sex, age, or weight. There was a low potential for drug-drug interactions, including low potential for interaction with the only other drug that was approved for ALS at that time. A review of natural history data was performed and found no significant racial differences in ALS diagnosis, progression, or biomarker data. And the medical practice for ALS was similar in both countries. So overall supporting FDA's determination of the drug being ethnically insensitive. So I'm actually currently working with a sponsor who is pursuing an ICHE5 strategy for approval of their drug in the US. They have good phase 3 data from an ex-US study, and there really haven't been any questions raised about the drug's sensitivity to intrinsic factors. The drug is a monoclonal antibody, so metabolic enzyme polymorphisms are not a concern, and there do not appear to be significant gender differences in drug response. There are some differences in age distribution in terms of disease prevalence as compared to the US population, but I think that could probably be overcome fairly easily if that was the only issue. FDA's greater concern, based on their communications with the sponsor, pertains to extrinsic factors. The clinical study was conducted in a low to moderate income country, which generally speaking has a lower standard of medical access to care than is typical in the US. There have also been questions raised about possible differences in disease pathophysiology and outcomes based on the literature. And there are questions about the severity of disease at the time of treatment that are difficult to address without US data. The sponsor believes that their drug is in fact ethnically insensitive and is continuing to make that case to FDA.
In this example that you're talking about, Kevin, did they have discussions with FDA on their approach to their use of foreign data from their trials?
In fact, yes, they did, which makes the situation a bit more frustrating for them because they discussed their approach at the time of their end of phase 1 meeting. I think a nuance there is that at that time, there was still some expectation that there would be at least a little bit of US data included in the application at the time they submitted their BLA. that plan changed over time to the point where it's now almost exclusively ex-US data that's included in their application. So there's probably some frustration on both sides there.
And Kevin, it's great to see the successful examples, but I think, collectively we've learned almost more from the less successful examples that have been publicly kind of disclosed over the past couple of years. So let's start with surufatinib, which was being pursued for for an indication for the treatment of pancreatic and extrapancreatic neuroendocrine tumors. At a May 2020 pre-NDA meeting, the FDA had indicated to the company that the planned 2 phase 3 clinical trials plus a US bridging study may be enough for approval. However, after full review of the data package upon submission, the current FDA opinion from the complete response letter from May of 22 indicated a need for additional clinical data before approval will be granted. The FDA specified that a multi-regional clinical trial is needed, and it should include those who are representative of the patient population in the US and aligned to current medical practice in the US, which this was one of the extrinsic factors that Kevin had talked about earlier in the presentation. The second example is sintilimab, which Kevin also mentioned earlier in our discussion today. For this product, the data was considered non-generalizable to the US population, with the complete response letter indicating the need for, again, a multi-regional clinical trial before US approval would be considered. There were concerns raised around the application's lack of pre-planned survival analysis, the primary endpoint of progression-free survival, and a lack of planned testing for overall survival, which is, folks who are familiar with oncology know is kind of the oncology gold standard endpoint for the most part. So this is a situation, I think, where upon maybe proper utilization of the ICH E5 decision tree that we reviewed previously in one of our slides, the answer to question one of does it meet the local regulatory standards where you intend to submit your product, the answer to that probably should have been no, indicating the need for more clinical work potentially with that product. And then it seems that the last example was actually successful in the utilization of ICH E5 methodology from a clinical perspective. However, the application could not be approved because of quality issues. So toripalimab would have been the first immunotherapy in the US indicated to treat advanced nasopharyngeal carcinoma. FDA had communicated in the complete response letter that the current clinical data provided in the BLA supports the application despite the fact that the clinical trial data originated primarily from China, Taiwan, and Singapore. And FDA went on to state that this indication warrants regulatory flexibility with respect to the sufficiency of single region clinical data. And it seems that part of what went into this conclusion was the fact that people of Asian ancestry have been found to have the highest nasopharyngeal carcinoma incidence and overall mortality rates in comparison with all other studied racial groups. So essentially, studies in this group of patients was very representative of the expected population that would be treated overall.
Thank you, Don. All right, so that actually brings us to the end of the content that we wanted to share with you all today. Just in closing, to review some of the key points, we want to acknowledge that ex-US clinical trials are an important element in many drug development programs. That is a trend that has been the case for many years now, and there's no indication that that's going to change anytime soon. There is a pathway for US approval solely using non-US data. It has been successfully used by a number of companies for a number of products, as we've discussed today. But that clinical data package must meet all U.S. regulatory standards for approval, and it must demonstrate that the data is suitable for extrapolation to the U.S. population. Part of that assessment and demonstration is evaluation of ethnic sensitivity and potentially use of bridging studies in order to demonstrate the generalizability of your data package to a U.S. population. Diverse clinical trial enrollment remains an important developmental consideration, though it may not be possible or relevant for all programs. And we do recommend that sponsors discuss the timing of their clinical program and their plans for use of ex-US data with FDA uh, early and often as you develop data. I think it's a good idea, even if you have discussed the topic with FDA in an early meeting, that you revisit that topic in your later development meetings to ensure that as you develop more information about the drug, to ensure that your plans for bridging and for um, demonstration of ethnic sensitivity are consistent with FDA's interpretation of your data as well. And of course, the goal there is to reduce duplication of clinical studies to the extent that that's feasible and scientifically appropriate. Uh, so that, as I mentioned earlier, FDA engagement can be done at different points of development, and it is critical to de-risk your program.
Great. Thank you, Kevin. We're going to move on to the Q&A portion of our presentation today. And while I look through the questions, Kevin, I do have one thing that I was thinking about kind of to get us started. How much does unmet medical need play into FDA's decisions for using foreign data?
Yeah, it's a good question. I think it has a significant influence. It's certainly not the only factor, and an unmet medical need, won't cure all ills, as they say. But the existence of significant unmet medical need in the U.S., where available therapy, or where there is no available therapy, or the available therapy is, just doesn't provide satisfactory treatment for patients, that does tend to influence FDA to provide additional regulatory flexibility and may increase their willingness to rely on ex-US data to support approval of a particular product.
Okay. One question we have here, are there countries or regions that FDA treats as equivalent to the US when assessing foreign data?
Yeah, I think yes, with some caveats. I think studies that are conducted in the ICH regions, so Europe, UK, um, Australia, Japan, those tend to be viewed by FDA in most cases as being reasonably reliable and potentially representative of the U.S. population. That said, there still can be differences in available therapies that are approved in one country or region that aren't available in another region, that can change the context of care, and so that element has to be considered. And I think there's also still the question of evaluating your racial diversity in your clinical study and determining whether that's whether you have adequately evaluated your drug in racial populations that are present in the U.S. that may not be present at the same level in those other countries.
Okay. Appreciate the insight on that, Kevin. We didn't have any other questions right now. I will remind everyone, feel free to submit questions to the chat. We will follow up after the presentation with anything else that comes in, and we want to thank everybody for their time and attention today.